Hello and welcome to Silent Science. I'm so glad you're here. Whether you're curled up in bed, lying on the couch, or gently swaying in a hammock somewhere far away, this video is your invitation to unwind. Tonight, we're taking a slow, soothing journey into the tiniest of places deep inside your very own cells to explore the mysterious and magnificent world of mitochondria. Now, I always aim for that perfect balance, interesting enough to make your brain smile, but not so fascinating that it leaves you wide-eyed at 2 a.m., reciting ATP production stages in your sleep. If it does keep you awake, I hope it's only because you're drifting down a rabbit hole of gentle curiosity and maybe learning something lovely along the way. Before we begin, I'd be ever so grateful if you could give this video a like and maybe subscribe to the channel. It really helps me keep the lights on and the mitochondria happy. And while you're settling in, feel free to let me know in the comments where in the world you're watching from and what time it is there. I'm always delighted to know where and when we're sharing this quiet corner of science together. This video is designed to be soft, slow, and thoughtful. There will be no sudden noises or flashy transitions, just a calm stroll through the inner workings of life's tiniest engines. And as always, please remember that while we'll be talking a bit about health and biology, this video is purely for relaxation and educational purposes. It is not medical advice. With that said, let's begin. A hidden world inside your imagine. You've been invited to tour a futuristic research lab, the kind that hums with soft lights and low, gentle beeps. There's glass everywhere, microscopes the size of dinner plates, and robotic arms gracefully moving petri dishes as though dancing to a silent song. At the very center of the room, behind the most secure doors, lies the focus of the entire facility: mitochondria. Not just ordinary mitochondria, though we're talking engineered, edited, supercharged mitochondria designed not only to function, but to thrive. This isn't just a matter of quantity. It's about timing, location, and precision. Mitochondria and neurons are highly mobile. They move along tiny cellular tracks, shuttled by motor proteins to exactly where they're needed most. If a part of the neuron is particularly active, a synapse that's firing rapidly, mitochondria are sent there like a team of emergency electricians, delivering a fresh supply of energy and tools. This dynamic positioning is crucial, especially during learning and memory formation. When you learn something new, your brain strengthens certain synaptic connections, and mitochondria rush in to support those changes, ensuring that the right neurons have the power they need to adapt and grow. But how do these microscopic structures know where to go? How do they respond to the body's ever-changing demands? The answers lie in an intricate system of signaling pathways, chemical messages, zipping around to tell the mitochondria what's going on. If you're exercising, those signals will push mitochondria to increase ATP production. If you're resting, they'll dial things down. If you're cold, they might ramp up a process called thermogenesis, which generates heat to help keep your body warm. One of the most fascinating examples of this is brown fat. Unlike the more familiar white fat, which stores energy, brown fat's main job is to burn energy to produce heat. Babies have a lot of it, helping them regulate their body temperature before they develop the ability to shiver. But adults have it too, and its activity is closely tied to mitochondrial function. In fact, scientists are exploring ways to activate brown fat as a potential treatment for obesity and metabolic disorders. Mitochondria don't just generate energy, they also play a role in cell death. Yes, even these tiny powerhouses participate in the grand cycle of life and decay. When a cell becomes damaged or diseased, mitochondria release specific proteins that trigger apoptosis, a programmed self-destruction mechanism that prevents harmful cells from spreading. Without this process, cancerous cells could proliferate unchecked. So, in a way, Mitochondria act as both life-givers and executioners, maintaining a delicate balance between survival and elimination. As we dive deeper into this hidden world, one thing becomes clear. Mitochondria are far more than just cellular batteries. They are ancient, strange, semi-autonomous entities with a story stretching back billions of years. Every cell in your body that needs energy, almost all of them relies on mitochondria to do the heavy lifting. Think of a muscle cell in your leg. It needs enormous amounts of energy to help you stand, walk, or run. So, it's packed with mitochondria. A liver cell is also energy hungry. Heart cells, absolutely bursting with them. And yet, despite their importance, mitochondria remain one of the most overlooked aspects of human biology. For decades, they were simply labeled the powerhouse of the cell, a phrase that barely scratches the surface of their complexity. Today, thanks to advances in genetic research and biochemistry, we're beginning to understand just how much mitochondria influence everything from aging to disease to even our moods. So, let's take a moment to appreciate these quiet workers within us. Let's imagine shrinking down to their level, peering into the microscopic machinery that keeps us alive. Because beneath the surface of every breath, every thought, every heartbeat, there's an unseen symphony playing out, one conducted by the tiny, tireless engines known as mitochondria. The ancient origins of mitochondria, if we rewind the clock far enough, long before humans walked the earth, before forests blanketed the land, even before the first single-celled organisms developed complex structures, there was a fateful encounter that would shape the course of life itself. Somewhere in the primordial oceans, an ancient bacterial cell drifted into a larger host's cell, not as prey, but as a guest. Instead of being digested, this invader found a place to stay, offering something valuable in return: a new, more efficient way to generate energy. Over time, this relationship became permanent, giving rise to one of the most pivotal evolutionary innovations in history, the mitochondrion. The evidence for this theory, known as the endosymbiotic hypothesis, is compelling. Mitochondria possess their own circular DNA, separate from the nuclear genome, much like bacteria. They replicate independently through a process similar to binary fission, just as prokaryotes do. Even their ribosomes, the molecular machines responsible for protein synthesis, bear a striking resemblance to those found in bacteria, rather than the ribosomes in the rest of the cell. These similarities suggest that mitochondria were once free-living organisms, likely relatives of modern-day alpha proteobacteria that merged with early eukaryotic cells over 1 billion years ago. This fusion was revolutionary. Prior to the arrival of mitochondria, cells relied on anaerobic metabolism, fermentation, to generate energy, a relatively inefficient process. But with mitochondria on board, cells gained access to a far superior method of energy production: aerobic respiration. By utilizing oxygen to extract maximum energy from nutrients, mitochondria enabled cells to grow larger, develop specialized functions, and eventually form multicellular organisms. Without this leap, the evolution of complex life, including plants, animals, and humans, would not have been possible. Even today, remnants of this ancient partnership persist. While most mitochondrial genes have been transferred to the nucleus, over the course of evolution, a small but essential set remains within the mitochondria themselves. This leftover genetic material, mitochondrial DNA, mtDNA, tells the story of their bacterial ancestry. It encodes key components of the electron transport chain, the biochemical pathway responsible for producing ATP, the universal energy currency of life. Yet, despite their deep integration into our biology, mitochondria still retain a degree of autonomy. They can divide independently, communicate with the rest of the cell through intricate signaling networks, and even undergo selective quality control processes to ensure their continued functionality. This semi-independent nature makes them both powerful allies and vulnerable targets if mitochondrial function is compromised. The consequences can ripple across the entire organism. By understanding the origins of mitochondria, we gain insight not only into the fundamental mechanisms of life, but also into the interconnectedness of all living things, from the simplest yeast cells to the most complex human tissues. Mitochondria serve as a living testament to an ancient alliance, one that continues to fuel life on Earth. To this day, the genetic legacy of mitochondria, most of the genetic material in your cells lives inside the nucleus, the big central hub that acts like a master archive. The nucleus contains all the instructions needed to make you, from the color of your eyes, to the shape of your nose, to how your cells should grow, divide, and respond to the world. But the mitochondria, they have their own miniature instruction manual. It's shorter, more compact, and written in an older dialect, but it's theirs, and it tells an ancient story. Mitochondrial DNA, or mtDNA for short, is circular and like the linear chromosomes in the nucleus and remarkably compact. While the nuclear genome contains roughly 20,000 genes, mitochondrial DNA holds only 37. That may seem like a small number, but those few genes punch well above their weight. 13 of them code for essential proteins involved in oxidative phosphorylation, the process by which mitochondria generates ATP. The remaining 24 genes are responsible for building transfer RNAs, tRNAs, and ribosomal RNAs, critical players in translating genetic information into functional proteins. Having their own genetic blueprint allows mitochondria to operate with a degree of independence. When energy demand spikes during exercise, for example, mitochondria can ramp up their own protein production without waiting for instructions from the nucleus. This local control ensures that energy production can respond swiftly to the cell's immediate needs. It's like having an emergency toolkit on hand, rather than relying solely on a distant command center. But this autonomy comes with trade-off. Unlike nuclear DNA, which benefits from multiple layers of repair mechanisms, mitochondrial DNA is more vulnerable to damage, because mitochondria are constantly generating reactive oxygen species. ROS is a byproduct of energy production. Their DNA is exposed to higher levels of oxidative stress. Over time, this can lead to mutations that impair mitochondrial function. In fact, accumulating mtDNA mutations have been linked to aging, neurodegenerative diseases, and metabolic disorders. Despite these risks, the presence of mitochondrial DNA is a powerful piece of evidence supporting the endosymbiotic origin of mitochondria. Their genetic makeup bears a striking resemblance to that of free-living bacteria, reinforcing the idea that they were once independent organisms. Today, they remain deeply integrated into our biology, carrying forward a legacy that stretches back over 1 billion years. By studying mitochondrial DNA, scientists have uncovered fascinating insights into human evolution, inheritance patterns, and even forensic identification. Because mtDNA is passed down almost exclusively from mother to child, it provides a direct line of descent through maternal ancestry. This unique inheritance pattern has allowed researchers to trace human migration patterns across continents, and even reconstruct the genetic lineage of our earliest ancestors. So, while the nucleus holds the bulk of your genetic information, don't underestimate the significance of the small but mighty mitochondrial genome. It's a relic of an ancient past, a blueprint for energy production, and a window into the deep history of life itself. Mitochondria, the architects of cellular energy. Let's picture a bustling factory, humming with activity. Conveyor belts carry raw materials into massive processing units, where skilled workers transform them into usable products. machines whir, pistons pump, and energy flows through every operation. Now shrink that entire industrial complex down to the microscopic scaling side. Your cells. Here, the factory is your body, and the mitochondria are the power plants keeping everything running smoothly. At the heart of mitochondrial function is ATP, pyadenosine triphosphate, the molecule that fuels nearly every biological process in your body. Without ATP, your muscles wouldn't contract, your nerves wouldn't fire, and your organs wouldn't function. Think of ATP as the universal currency of energy, and mitochondria are the minting machines that produce it. The process begins with the food you eat. Carbohydrates, fats, and proteins are broken down into smaller molecules, which enter the mitochondria to be further processed. Glucose, derived from carbohydrates, is split into pyruvate in the cytoplasm through glycolysis. Pyruvate then enters the mitochondrial matrix, where it undergoes the Krebs cycle, also known as the citric acid cycle. During this multi-step process, carbon atoms from pyruvate are gradually stripped away, releasing electrons that will be used to generate ATP. But the real magic happens in the electron transport chain, a series of protein complexes embedded in the inner mitochondrial membrane. As electrons pass through these complexes, their energy is harnessed to pump protons across the membrane, creating a gradient. This proton gradient is like water held behind a dam, potential energy waiting to be released. When protons flow back through a protein called ATP synthase, that energy is converted into ATP, much like water turning a turbine to generate electricity. This process, known as oxidative phosphorylation, is astonishingly efficient. A single glucose molecule can yield up to 36 ATP molecules when fully metabolized in the mitochondria, far more than the 2 ATP produced in glycolysis alone. And it's not just glucose. Fatty acids and amino acids can also be broken down to fuel ATP production, allowing mitochondria to adapt to different dietary conditions. But mitochondria don't just respond to the body's energy needs, they anticipate them. If you start running, your muscles require more energy, and mitochondria ramp up ATP production accordingly. If you're fasting, they switch to burning fat instead of carbohydrates. This flexibility is crucial for survival, enabling the body to maintain energy homeostasis under varying conditions. Beyond energy production, mitochondria play a role in regulating metabolism, calcium signaling, and even programmed cell death. They are dynamic structures, constantly fusing and dividing to optimize function, repair damage, and distribute energy efficiently throughout the cell. So, next time you take a breath, remember that each inhalation delivers oxygen to your mitochondria, fueling the intricate dance of molecules that keeps you alive. From the moment you wake up to the second you drift off to sleep, your mitochondria are hard at work, quietly powering your existence, one molecule at a time. The dynamic life of mitochondria are not static power plants confined to a single location within the cell. Instead, they are constantly on the move, navigating the intricate landscape of the cytoplasm like tiny autonomous vehicles. This mobility is essential for maintaining cellular function, ensuring that energy is delivered precisely where and when it is needed most. In neurons, for example, where energy demands fluctuate rapidly based on synaptic activity, mitochondria must travel along axons and dendrites to reach active regions. Motor proteins act as couriers, shuttling mitochondria along microtubule highways to their destinations. Once they arrive, mitochondria engage in a continuous process of fusion and fission to opposing mechanisms that allow them to adapt to changing conditions. Fusion enables mitochondria to merge, exchanging contents and repairing damaged components. This blending of resources ensures that mitochondria can maintain optimal function, diluting harmful mutations and preserving genetic integrity. Conversely, fission allows mitochondria to divide, separating damaged sections for removal, while generating new, healthy mitochondria. This balance between fusion and fission is crucial for mitochondrial quality control, preventing the accumulation of dysfunctional organelles that could compromise cellular health. These dynamic behaviors extend beyond mere maintenance. Mitochondria also play a role in intercellular communication, influencing gene expression and signaling pathways that affect overall physiology. When mitochondria detect shifts in energy demand, they send molecular signals to the nucleus, prompting the cell to adjust its metabolic strategy. This dialogue ensures that energy production aligns with the body's needs, whether during intense physical activity, periods of fasting, or times of rest. Furthermore, mitochondria contribute to cellular resilience by managing stress responses. Under challenging conditions, such as exposure to toxins, oxidative stress, or nutrient scarcity, mitochondria initiate protective measures. They activate antioxidant defenses, modulate calcium signaling, and even trigger autophagy, the process by which damaged cellular components are recycled. These adaptive responses highlight mitochondria's role, not only as energy producers, but also as guardians of cellular stability. Understanding the fluid, responsive nature of mitochondria reveals their profound impact on health and disease. Disruptions in mitochondrial dynamics have been linked to neurodegenerative disorders, metabolic syndromes, and age-related decline. By maintaining a delicate equilibrium between movement, fusion, fission, and signaling, mitochondria uphold the intricate balance necessary for life. Mitochondria and the brain. A delicate partnership. Mitochondria and the brain are partners in a very complex dance. One provides the energy, the other directs the moves. When the rhythm is right, the brain thrives, thinking clearly, feeling deeply, remembering richly. When that rhythm falters, when mitochondria can't keep up, the entire performance begins to wobble. But the beauty of it is this: every healthy habit, every nourishing choice, every moment of rest or movement or laughter, it all supports the dance. It helps keep the mitochondria in your brain flexible, fueled, and ready to respond. Your brain consumes an extraordinary amount of energy, about 20% of your total resting energy expenditure, despite making up only about 2% of your body weight. Neurons are incredibly active cells, firing electrical signals, maintaining ion gradients, recycling neurotransmitters, and forming new connections, all of which demand a steady supply of ATP. Without mitochondria, these processes would grind to a halt. Consider a simple action, like reading this sentence. Light enters your eyes, stimulating photoreceptor cells that convert visual input into electrical signals. Those signals travel through complex neural pathways, requiring rapid bursts of energy to sustain the electrochemical reactions that allow perception and comprehension. Mitochondria within your neurons ensure that ATP is continuously generated to support this activity, beyond basic cognitive function. Mitochondria also play a crucial role in mood regulation. Neurotransmitters such as serotonin, dopamine, and norepinephrine rely on mitochondrial energy to be synthesized, released, and reabsorbed. Imbalances in these chemical messengers are associated with depression, anxiety, and other mental health disorders. Emerging research suggests that mitochondrial dysfunction may contribute to these conditions, highlighting the deep connection between cellular energy production and emotional well-being. Learning and memory formation are particularly dependent on mitochondrial efficiency. When you acquire new knowledge, your brain strengthens specific synaptic connections through a process known as long-term potentiation, LTP. This requires a surge of energy, and mitochondria are instrumental in supplying that power. Studies have shown that impaired mitochondrial function can hinder LTP, leading to difficulties in forming and retaining memories. Stress also influences mitochondrial-brain interactions. Short-term stress can actually enhance mitochondrial function through hormesis, phenomenon where mild stresses trigger adaptive responses that strengthen cells. Exercise, for instance, temporarily increases oxidative stress, prompting mitochondria to boost their antioxidant defenses and improve efficiency. However, chronic stress tells a different story. Prolonged exposure to elevated cortisol levels disrupts mitochondrial signaling, reduces energy availability, and contributes to cognitive fatigue and mood disturbances. Supporting mitochondrial health in the brain involves a combination of lifestyle factors. Regular physical activity promotes mitochondrial biogenesis, increasing both the number and efficiency of these energy-producing structures. Nutrient-rich diets, particularly those containing antioxidants and omega-3 fatty acids, provide the building blocks necessary for optimal mitochondrial function. Quality sleep allows mitochondria to perform essential repair processes, clearing out damaged components and restoring cellular balance. The relationship between mitochondria and the brain is a reminder that mental and physical health are deeply intertwined. Every choice you make, what you eat, how you move, how you manage stress echoes within the microscopic world of your cells, shaping the foundation of your thoughts, emotions, and memories. Taking care of your mitochondria isn't just about longevity. It's about ensuring that your mind remains sharp, resilient, and full of wonder. Supporting mitochondrial health, lifestyle choices that make a difference. Every healthy habit, every nourishing choice, every moment of rest or movement, or laughter, all supports the dance of life orchestrated by your mitochondria. These microscopic powerhouses respond beautifully to thoughtful care, adapting to your lifestyle choices and enhancing your overall well-being. The good news is that supporting mitochondrial health doesn't require exotic interventions or expensive supplements. It starts with the everyday decisions you make, choices that, when combined, create a powerful foundation for cellular vitality. Regular physical activity is one of the most effective ways to boost mitochondrial function. Exercise stimulates mitochondrial biogenesis, the process by which new mitochondria are created, ensuring that your cells have ample energy to meet their demands. Aerobic activities like walking, swimming, or cycling increase oxygen delivery to tissues, promoting endurance and enhancing mitochondrial efficiency. Resistance training builds muscle mass, which in turn increases the number of mitochondria in skeletal muscle cells. High-intensity interval training height, alternating between short bursts of intense effort and recovery periods, has been shown to significantly improve mitochondrial capacity in a relatively short time. Even modest movement, such as taking the stairs instead of the elevator or going for a short walk, sends positive signals to your mitochondria, encouraging them to become stronger and more efficient. Nutrition also plays a crucial role in mitochondrial health. A diet rich in whole foods, especially colorful fruits and vegetables, provides essential vitamins, minerals, and antioxidants that protect mitochondria from oxidative stress. Certain nutrients, such as coenzyme Q10, COQ10, carnitine, and B vitamins, are directly involved in mitochondrial energy production. Omega-3 fatty acids, found in fish, flax seeds, and walnuts, support mitochondrial membrane integrity, while polyphenols from sources like green tea, dark chocolate, and berries enhance mitochondrial function through anti-inflammatory effects. Additionally, emerging research suggests that intermittent fasting and time-restricted eating may benefit mitochondria by activating cellular repair mechanisms and improving metabolic flexibility. Sleep is another pillar of mitochondrial well-being. During rest, your body engages in critical repair processes, including mitochondrial maintenance. Deep sleep cycles promote the clearance of damaged proteins and the regeneration of cellular structures, ensuring that mitochondria function optimally. Chronic sleep deprivation, on the other hand, disrupts mitochondrial signaling, leading to reduced energy production and increased oxidative stress. Prioritizing consistent high-quality sleep allows your mitochondria to recharge, reinforcing their ability to support cognitive function, mood regulation, and overall resilience. Stress management completes the trifecta of mitochondrial support. While moderate stressors like exercise and fasting can be beneficial triggering adaptive responses that strengthen mitochondria, chronic psychological stress has the opposite effect. Prolonged exposure to stress hormones like cortisol impairs mitochondrial function, reducing energy availability and contributing to fatigue, brain fog, and immune disruption. Mindfulness practices such as meditation, deep breathing, and yoga help mitigate these effects by promoting cellular balance and enhancing mitochondrial resilience. By incorporating these lifestyle strategies into your daily routine, you provide your mitochondria with the tools they need to thrive. Each small choice, whether it's choosing a nutrient-dense meal, taking a walk, getting enough rest, or finding moments of calm, contributes to the greater symphony of health, ensuring that your cellular powerhouses continue to fuel your body and mind with unwavering energy. The future of mitochondrial medicine as scientific understanding of mitochondria deepens. So too does the potential for groundbreaking medical advancements. Researchers are now exploring innovative ways to harness mitochondrial function, not just to treat disease, but to prevent it at its very core. Gene therapy, mitochondrial transplantation, and targeted pharmaceutical interventions are emerging as promising avenues for addressing mitochondrial dysfunction, offering hope for conditions ranging from neurodegenerative disorders to metabolic syndromes. One of the most exciting developments is the use of gene editing technologies to correct mitochondrial DNA mutations. Mitochondrial diseases, often inherited and difficult to treat, result from defects in mtDNA that impair energy production. Recent advancements in techniques like CRISPR, Cas9, and base editing have opened the door to precise modifications of mitochondrial genes, potentially correcting mutations that cause debilitating conditions such as Leigh syndrome, mitochondrial encephalomyopathy, and Leber's hereditary optic neuropathy. While challenges remain due to the unique structure and replication mechanisms of mitochondrial DNA, progress in this field suggests that future therapies may be able to restore mitochondrial function at the genetic level. Beyond gene therapy, mitochondrial transplantation is gaining traction as a novel approach to treating tissue damage and organ failure. In experimental settings, scientists have successfully transferred healthy mitochondria into cells with impaired function, effectively rescuing energy production and cellular viability. This technique holds promise for conditions such as heart disease, stroke, and acute lung injury, where mitochondrial dysfunction plays a critical role in tissue damage. Clinical trials are currently investigating the feasibility of mitochondrial transplantation in human patients, with early results indicating potential benefits in cardiac and neurological recovery. Pharmaceutical interventions targeting mitochondrial function are also advancing rapidly. Compounds designed to enhance mitochondrial efficiency, reduce oxidative stress, and support metabolic pathways are being tested for their therapeutic potential. One area of interest is the development of NAD boosters, which aim to replenish declining levels of nicotinamide, adenine dinucleotide NAD, a coenzyme essential for mitochondrial energy production and DNA repair. Supplements such as nicotinamide, riboside NR, and nicotinamide mononucleotide NMN are being studied for their ability to improve mitochondrial function in aging populations and individuals with metabolic disorders. Additionally, researchers are investigating the role of mitochondrial-targeted antioxidants, such as Mitoch and SKQ1, which neutralize harmful reactive oxygen species, ROS, more effectively than conventional antioxidants. These compounds have shown promise in studies for conditions like Parkinson's disease, Alzheimer's disease, and cardiovascular disorders, suggesting that protecting mitochondria from oxidative damage may slow the progression of degenerative diseases. As these cutting-edge therapies continue to evolve, ethical considerations surrounding mitochondrial manipulation come into focus. Questions about safety, accessibility, and long-term effects must be carefully addressed before widespread implementation. Nevertheless, the growing field of mitochondrial medicine represents a paradigm shift in healthcare, one that prioritizes cellular health as the foundation for overall well-being. With each discovery, we move closer to a future where mitochondrial dysfunction is not just managed, but actively corrected. The implications extend beyond disease treatment, touching on the very essence of human longevity, resilience, and adaptation. As science continues to unlock the mysteries of these microscopic powerhouses, the possibilities for transforming medicine, ultimately, life itself, far boundless. The rhythms of mitochondrial adaptation. Mitochondria are not static energy generators. They are dynamic structures that constantly adjust to the body's shifting needs. This adaptability is evident in how they respond to fluctuations in energy demand, environmental stresses, and metabolic signals. Just as a musician fine-tunes an instrument to match the tempo of a symphony, mitochondria modulate their function to maintain cellular harmony. One of the most remarkable aspects of mitochondrial responsiveness is their ability to alter their metabolic preferences based on available fuel sources. While glucose is a primary energy substrate, mitochondria can seamlessly switch to using fatty acids or ketones when carbohydrate availability is limited. This metabolic flexibility is crucial for survival, especially during periods of fasting or prolonged exercise when glycogen stores are depleted. The transition from glucose to fat-based metabolism is orchestrated by a network of signaling pathways, including AMP-activated protein kinase, AMPK, and peroxisome proliferator activated receptors, PPARs, which regulate gene expression related to mitochondrial function. Exercise serves as a prime example of how mitochondria adapt to increased energy requirements. During physical activity, muscle cells experience heightened ATP consumption, prompting mitochondria to ramp up energy production. This surge in demand triggers mitochondrial biogenesis, the creation of new mitochondria through the activation of transcription factors such as PGC1 alpha, peroxisome proliferator receptor gamma coactivator 1 alpha. Over time, regular exercise enhances mitochondrial density, improves metabolic efficiency, and boosts overall cellular resilience. Temperature regulation is another domain where mitochondrial adaptability shines. When exposed to cold, mitochondria in brown adipose tissue, BAT activate thermogenesis, a process that generates heat by uncoupling oxidative phosphorylation. This means that instead of producing ATP, the energy from nutrient breakdown is dissipated as warmth, helping maintain body temperature in chilly environments. The key player in this process is uncoupling protein 1-UCP1, which allows protons to bypass ATP synthase, effectively converting stored energy into heat. This mechanism is particularly vital in newborns, who lack the ability to shiver and rely heavily on BAT for thermoregulation. Mitochondria also exhibit remarkable plasticity in response to dietary changes. Intermittent fasting and caloric restriction have been shown to enhance mitochondrial function by activating cellular stress responses that promote repair and longevity. These dietary interventions stimulate mitophagy, the selective degradation of damaged mitochondria, ensuring that only the most efficient organelles remain. Additionally, fasting-induced metabolic shifts encourage the use of ketones as an alternative fuel source, which has been linked to improved cognitive function and neuroprotection. Beyond external influences, mitochondria engage in constant communication with the rest of the cell through intricate signaling pathways. Calcium ions, reactive oxygen species, ROS, and various metabolites act as molecular messengers, relaying information about energy status and stress levels. When cellular stress increases, mitochondria release signals that prompt the nucleus to upregulate antioxidant defenses, enhance DNA repair mechanisms, and modulate inflammatory responses. This bidirectional communication ensures that mitochondria remain attuned to the broader physiological landscape, adjusting their behavior to maintain cellular equilibrium. The rhythmic adaptability of mitochondria underscores their role as more than just energy producers. They are sophisticated regulators of cellular homeostasis. By responding to metabolic cues, environmental stimuli, and internal signals, mitochondria ensure that energy production remains finely tuned to the body's ever-changing demands. Mitochondria and aging. The longevity connection. Aging is one of those strange, universal experiences, something we all share but don't fully understand. It creeps in slowly, almost politely at first. A grey hair here, a slight stiffness in the joints there. But beneath these visible signs lies a deeper, more intricate process, one that unfolds at the cellular level, where mitochondria play a central role. As we grow older, mitochondrial function tends to decline, and this gradual deterioration is increasingly recognized as a key driver of aging and age-related diseases. At the heart of this connection is the accumulation of mitochondrial DNA mutations. Unlike nuclear DNA, which benefits from robust repair mechanisms, mtDNA lacks the same level of protection and fidelity. Over time, errors in mtDNA replication accumulate, leading to impaired mitochondrial function. Some of these mutations disrupt the electron transport chain, reducing the efficiency of ATP production and increasing the generation of reactive oxygen species, ROS. ROS, while naturally produced during energy metabolism, can become problematic when they overwhelm the cell's antioxidant defenses. Excessive ROS leads to oxidative stress, damaging cellular components such as proteins, lipids, and even nuclear DNA. This creates a vicious cycle. Damaged mitochondria produce more ROS, which in turn accelerates cellular aging. Another factor contributing to mitochondrial aging is the decline in mitophagy, the process by which damaged mitochondria are selectively removed and recycled. In younger cells, mitophagy efficiently clears out dysfunctional mitochondria, ensuring that only the healthiest organelles remain. However, as we age, this quality control mechanism becomes less efficient. Damaged mitochondria accumulate further, compromising cellular energy production and increasing inflammation. This decline in mitochondrial turnover has been linked to numerous age-related conditions, including neurodegenerative diseases like Parkinson's and Alzheimer's, as well as metabolic disorders such as type 2 diabetes. Interestingly, some research suggests that mitochondrial dysfunction may not only be a consequence of aging, but also a contributor to its progression. Studies on model organisms, such as nematodes and fruit flies, have shown that interventions that enhance mitochondrial functions, such as calorie restriction, exercise, or supplementation with mitochondrial supporting compounds, can extend lifespan and improve health span. In mammals, similar findings have emerged, boosting mitochondrial biogenesis through activators like PGC1 alpha has been associated with improved metabolic function, and delayed onset of age-related diseases. One intriguing aspect of mitochondrial aging is its connection to the immune system. As mitochondria degrade, they release fragments of their own DNA into the cytoplasm. This mitochondrial DNA, mtDNA, can be mistaken by the immune system as foreign viral DNA, triggering inflammatory responses. This phenomenon, known as mitochondrial-derived inflammation or mitoinflammation, has been implicated in chronic inflammatory conditions associated with aging, such as atherosclerosis and autoimmune disorders. Understanding how mitochondria influenced immune signaling could open new avenues for combating age-related inflammation and extending healthy lifespan. While aging remains an inevitable part of life, the study of mitochondrial biology offers promising insights into how we might slow its effects. By supporting mitochondrial health through lifestyle choices, regular exercise, balanced nutrition, adequate sleep, and stress management, we can help preserve cellular energy production and delay the onset of age-related decline. Furthermore, ongoing research into mitochondrial-targeted therapies, such as gene editing, mitochondrial transplantation, and small molecule interventions, holds the potential to revolutionize how we approach aging, not as an unavoidable fate, but as a process that can be influenced and optimized. The evolutionary significance of mitochondria. Some scientists go even further and argue that mitochondria were the key requirement for the evolution of intelligence. After all, the human brain is a notorious energy hog. It makes up only about 2% of your body weight, but it uses around 20% of your resting energy supply. That's a lot of ATP just to think, dream, and occasionally forget where you put your phone. Without mitochondria providing that energy, a brain like ours simply wouldn't be possible. Neurons are incredibly active cells, firing electrical signals, maintaining ion gradients, recycling neurotransmitters, and forming new connections, all of which demand a steady supply of ATP. Consider the cognitive leaps that distinguish humans from other species. Our ability to solve complex problems, create language, and build civilizations hinges on the brain's immense energy requirements. Mitochondria, with their unparalleled efficiency in generating ATP, made this possible. Early hominins with enhanced mitochondrial function likely had an advantage in cognitive development, enabling better problem-solving skills, improved memory, and more advanced social behaviors. This evolutionary edge may have played a crucial role in the emergence of Homo sapiens as the dominant species on Earth. Beyond intelligence, mitochondria also contributed to the development of complex multicellular organisms. Before the advent of mitochondria, cells relied on fermentation for energy production, a process that yields only 2 ATP molecules per glucose molecule. In contrast, mitochondrial respiration produces up to 36 ATP molecules per glucose, drastically increasing the energy available to cells. This surplus of energy allowed cells to specialize, giving rise to diverse tissues and organs. Without mitochondria, the evolution of complex life forms from plants and fungi to animals would not have been feasible. The evolutionary impact of mitochondria extends beyond energy production. Their presence facilitated the development of cellular differentiation, a cornerstone of multicellularity. With abundant energy, cells could afford to take on specialized roles, some dedicated to movement, others to reproduction, defense, or sensory perception. This specialization laid the groundwork for the vast diversity of life we see today. Moreover, mitochondrial signaling pathways helped coordinate cellular communication, ensuring that different tissues worked in harmony. Mitochondria also played a role in shaping the immune system. The release of mitochondrial DNA into the cytoplasm during cellular stress or damage can trigger immune responses, acting as an early warning system against infection or injury. This mechanism, believed to have evolved from the ancient bacterial origins of mitochondria, highlights their dual role as both energy producers and immune regulators. From the smallest single-celled organisms to the most complex brains in the animal kingdom, mitochondria have been silent architects of life's evolution. Their ability to generate energy efficiently, support cellular specialization, and contribute to immune defense has made them indispensable to the development of intelligent, complex life. As we continue to uncover the depths of mitochondrial biology, we gain not only a deeper appreciation for their role in our own existence, but also a glimpse into the evolutionary forces that shaped the natural world, mitochondria and disease. Unraveling the connections, mitochondrial dysfunction has been implicated in a wide range of diseases, from rare genetic disorders to common chronic conditions. Because mitochondria are central to energy production, cellular signaling, and stress response, any disruption in their function can have cascading effects throughout the body. Among the most well-known mitochondrial diseases are inherited disorders, caused by mutations in mitochondrial DNA, mtDNA, or nuclear genes encoding mitochondrial proteins. These conditions, such as Leigh syndrome, mitochondrial encephalomyopathy, and Leber's hereditary optic neuropathy, manifest in diverse ways, often affecting tissues with high energy demands, such as the brain, muscles, and heart. Beyond these, primary mitochondrial diseases, mounting evidence suggests that mitochondrial dysfunction plays a significant role in many common illnesses. Neurodegenerative diseases, like Parkinson's and Alzheimer's, for instance, show strong links to mitochondrial impairment. In Parkinson's disease, mutations in genes such as PINK1 and PARKIN, both involved in mitochondrial quality control, lead to the accumulation of damaged mitochondria in dopaminergic neurons. This dysfunction disrupts energy production and increases oxidative stress, contributing to neuronal death and the progressive loss of motor function. Similarly, in Alzheimer's disease, mitochondrial abnormalities are observed early in disease progression, with impaired respiratory chain activity and altered calcium signaling, exacerbating the buildup of toxic protein. Aggregates like beta and tau. Metabolic disorders, including type 2 diabetes and obesity, also intersect with mitochondrial health. Insulin resistance, a hallmark of type 2 diabetes, has been linked to mitochondrial dysfunction in muscle and fat cells. When mitochondria fail to efficiently oxidize fatty acids, excess lipids accumulate, interfering with insulin signaling and glucose uptake. This metabolic imbalance not only contributes to diabetes, but also increases the risk of cardiovascular complications. Likewise, in obesity, mitochondrial dysfunction in adipose tissue and the liver leads to impaired energy metabolism, promoting fat storage and systemic inflammation. Cardiovascular diseases are another area where mitochondrial dysfunction plays a crucial role. The heart, an organ with relentless energy demands, relies heavily on mitochondrial ATP production. In heart failure, mitochondrial inefficiency leads to reduced contractile function, as weakened mitochondria struggle to meet the energy needs of cardiac muscle cells. Ischemic heart disease, characterized by restricted blood flow, causes mitochondrial damage due to oxygen deprivation, followed by a surge of reactive oxygen species ROS upon reperfusion. This oxidative stress worsens tissue injury and impairs recovery after heart attacks. Autoimmune and inflammatory conditions also exhibit mitochondrial involvement. Mitochondrial DNA fragments released during cellular stress can act as damaging molecular patterns, DAMPs, triggering immune responses that contribute to chronic inflammation. In rheumatoid arthritis, for example, mitochondrial dysfunction in synovial cells promotes excessive ROS production and inflammatory cytokine release, exacerbating joint damage. Similarly, in lupus, mitochondrial abnormalities are linked to the misrecognition of self-DNA, leading to autoimmune reactions. Emerging research is also uncovering mitochondrial connections in psychiatric and neurodevelopmental disorders. Depression, bipolar disorder, and schizophrenia have all been associated with altered mitochondrial function, particularly in brain regions responsible for mood regulation and cognition. Mitochondrial dysfunction in these areas affects neurotransmitter synthesis, synaptic plasticity, and neuronal resilience, contributing to the symptoms of these conditions. Autism spectrum disorder, ASD, has also been linked to mitochondrial abnormalities, with studies showing impaired energy metabolism and increased oxidative stress in affected individuals. As research progresses, the intricate relationships between mitochondrial dysfunction and disease become increasingly apparent. These findings underscore the importance of maintaining mitochondrial health, not only for preventing rare genetic disorders, but also for mitigating the burden of chronic illnesses that affect millions worldwide. By understanding how mitochondrial dysfunction contributes to disease, scientists and clinicians can develop targeted therapies aimed at restoring mitochondrial function and improving patient outcomes. The interconnected web of mitochondria do not exist in isolation. They are deeply embedded in the intricate web of cellular communication, influencing and being influenced by virtually every biological process. Their role extends far beyond energy production, reaching into the realms of immunity, inflammation, and even cellular decision-making. One of the most fascinating aspects of mitochondrial biology is their ability to act as signaling hubs, constantly monitoring the cell's environment and responding to changes in metabolic state, stress levels, and immune threats. In the immune system, mitochondria serve as both defenders and communicators. Immune cells, particularly macrophages and T cells, rely heavily on mitochondrial function to mount effective responses against pathogens. When a virus or bacterium invades the body, immune cells rapidly shift their metabolism to meet the heightened energy demands of fighting infection. This metabolic reprogramming is orchestrated by mitochondria, which adjusts their activity to support rapid proliferation and effective functions. Beyond energy production, mitochondria contribute to antiviral defense by producing interferons and other signaling molecules that alert neighboring cells to the presence of an infection. Mitochondria also play a crucial role in the regulation of inflammation. When cells experience damage or infection, mitochondrial components, particularly mitochondrial DNA, mtDNA, and reactive oxygen species ROS, are released into the cytoplasm. These molecules act as danger signals, activating inflammatory pathways such as the inflammasome, a complex of proteins that triggers the release of pro-inflammatory cytokines. While this response is essential for eliminating threats, excessive or uncontrolled mitochondrial-driven inflammation can contribute to chronic diseases such as rheumatoid arthritis, atherosclerosis, and neurodegenerative disorders. Understanding how mitochondria influence inflammation opens new avenues for therapeutic intervention, with researchers exploring ways to modulate mitochondrial signaling to reduce pathological inflammation without compromising immune defenses. Another remarkable feature of mitochondrial biology is their involvement in programmed cell death, or apoptosis. When a cell sustains irreparable damage, whether from DNA mutations, oxidative stress, or infection, mitochondria initiate a controlled self-destruction process to prevent harm to surrounding tissues. This is achieved through the release of cytochrome C and other proapoptotic proteins, which activate caspases, the enzymes responsible for dismantling the cell in an orderly manner. Dysregulation of mitochondrial apoptosis has been implicated in cancer, where defective apoptotic pathways allowed damaged cells to survive and proliferate unchecked. Targeting mitochondrial apoptotic mechanisms is therefore a promising strategy in cancer therapy, aiming to restore the cell's ability to eliminate malignant cells before they spread. Even in the realm of cellular aging, mitochondria exert their influence. The accumulation of mitochondrial dysfunction over time contributes to senescence, a state in which cells cease to divide but remain metabolically active, often secreting inflammatory molecules that affect neighboring cells. This phenomenon, known as the senescence-associated secretory phenotype SASP, is linked to age-related diseases and tissue degeneration. By modulating mitochondrial function, researchers are investigating ways to delay cellular senescence and promote healthier aging through their roles in immunity, inflammation, apoptosis, and aging. Mitochondria emerge not just as powerhouses of the cell, but as central regulators of life and death. Their ability to sense and respond to cellular stress, coordinate immune defenses, and influence disease progression underscores their profound impact on human health. As our understanding of mitochondrial biology deepens, so too does our appreciation for the vast, interconnected networks that govern life at the most fundamental level. The future of mitochondrial research and therapeutic potential. As we look ahead, the field of mitochondrial research stands on the brink of transformative discoveries that could reshape medicine and human health. Scientists are now delving into previously uncharted territories, exploring how mitochondria interact with the microbiome, influence epigenetic regulation, and even contribute to regenerative medicine. One particularly promising avenue is the development of mitochondrial-targeted therapies that could slow aging, prevent chronic diseases, and enhance cellular resilience. Recent studies have begun to uncover the intricate relationship between mitochondria and the gut microbiome. The trillions of microbes residing in the digestive tract produce metabolites that influence mitochondrial function, either supporting energy production or inducing stress responses. For instance, short-chain fatty acids like butyrate, generated by beneficial gut bacteria, have been shown to enhance mitochondrial efficiency and reduce inflammation. On the flip side, dysbiosis and imbalance in microbial communities can lead to mitochondrial dysfunction, contributing to metabolic disorders, neurodegeneration, and immune dysregulation. Understanding these interactions opens the door to novel interventions such as probiotics, prebiotics, and dietary modifications that could optimize mitochondrial health through microbial modulation. Epigenetics, the study of gene expression changes without altering DNA sequence, also intersects with mitochondrial biology. Emerging research indicates that mitochondrial signals can influence chromatin remodeling and histone modifications, thereby affecting nuclear gene expression. This bidirectional communication between mitochondria and the nucleus suggests that mitochondrial health may play a role in developmental programming, stress adaptation, and even transgenerational inheritance. Scientists are investigating whether mitochondrial-derived metabolites, such as acetyl-CoA and NAD+, serve as epigenetic regulators, offering potential strategies for reversing disease-associated gene silencing or activation. Regenerative medicine is another frontier where mitochondria are proving to be game changers. Stem cell therapies, which hold promise for tissue repair and organ regeneration, rely heavily on mitochondrial function. Healthy mitochondria are essential for stem cell differentiation, proliferation, and engraftment. Researchers are experimenting with mitochondrial augmentation techniques, introducing healthy mitochondria into stem cells before transplantation to enhance their regenerative capacity. Additionally, mitochondrial transfer between cells, a naturally occurring process in some tissues, is being explored as a means to rescue damaged cells in conditions like myocardial infarction, spinal cord injury, and pulmonary disease. The convergence of artificial intelligence and systems biology is accelerating mitochondrial research, allowing scientists to model complex metabolic networks and predict how mitochondrial perturbations affect whole-body physiology. Machine learning algorithms are being trained on vast datasets to identify biomarkers of mitochondrial dysfunction, paving the way for early diagnosis and personalized treatment strategies. Meanwhile, synthetic biology approaches are being used to engineer mitochondria with enhanced capabilities, such as improved energy efficiency or resistance to oxidative stress. These advancements hint at a future where mitochondrial engineering could be tailored to individual needs, optimizing health. At the cellular level, as the boundaries of mitochondrial science expand, so too does the potential for groundbreaking medical applications. From microbiome-mitochondria interactions to AI-driven metabolic modeling, the next era of mitochondrial research promises to unlock new dimensions of human biology. What was once viewed as a simple cellular power plant is now understood to be a dynamic, multifaceted entity with far-reaching implications for health, longevity, and disease prevention. The journey into the microscopic world of mitochondria is far from complete, but every step forward brings us closer to harnessing their full potential for the benefit of humanity.